switching it out these days.
For Darwin Day. Let's see if this works. Yeah, this my laptop wasn't too compatible, so we're using John's in a pinch, and the cursor looks like a hand, and some slides didn't come in, but aside from those, you predict always technical problems, we're still going to manage pretty well, I hope. Well, here's the book that started it all. This is by a guy called Brian Sykes, Seven Daughters of Eve, here, and published in, I think it was 2001. Double check. Anyway, that's the book. So who was Brian Sykes? Yeah, here's a picture of Brian Sykes in his lab holding a bone, and he's looking at a bone. So why is Brian Sykes looking at a bone? Well, he started, let's go back to the 80s. He was a professor of genetics at Oxford, and he was examining something called lethal osteogenesis imperfecta, which is brittle bone disease, which is very sad. Your bones don't fully form, and they're very brittle, and you get lots of breaks. He was trying to find the genetic fault that caused brittle bone disease. Now, Oxford is right here. Here's London. Here's Oxford in England. Here's Oxford as a blow up. And there's a little town here called Abington, about 6 miles south of Abington. They were excavating for a supermarket, and they found they were on top of a medieval cemetery. So by the way they do things in England, they had stopped construction, and the archaeologists get 2 months to look at what the artifacts and what they can find down there. So they got some, these are like, this is medieval, this is about 1200s, your 750-year-old bones in the cemetery. And since Brian, who was at Oxford, had heard about this and he was interested in bones, they gave him some of the bones from a grave, and he was interested in see what can he do with the DNA in ancient bones. Well, to start with, here's DNA. Here's the classic double-strand helix. So what DNA are they going to look at? It turns out there are 2 types of DNA. Here's the irregular chromosomal DNA, your somatic DNA in your cell, in the cell nucleus. You've got about, well, 6 billion bases in a diploid number. It's big and it's bulky. And back in the 80s, it was hard to work with. They didn't have techniques for looking at it. But in your mitochondria, there's a little loop, a little circle of DNA called mitochondrial DNA, which seemed to be, probably ought to be easier to look at. Now, mitochondria are what they call the energy producers of the cell. That's where oxidation happens, where you make all your ATPs so you can move your arms and things. And you've got about 1,000 of them in every cell. So that's like, so there's 1,000 times more copies. They're smaller. There are maybe quite a few inside each mitochondrion. So that looked like that would be a better genes to look at. Whoops, let's go up. Now, first they tried cloning ancient DNA, but certainly at that time, ancient DNA did not clone very well. It's broken up into fragments, has oxidation damage. You just couldn't get it. So the idea in cloning, you take your favorite piece of DNA, put it in a plasmid, ligate it all together, put it in the bacteria, nice and easy, grow up the bacteria, and you've got lots of DNA. But it just wouldn't work. You couldn't clone stuff into old DNA into bacteria. Techniques have improved now and they can do it, but not then. But at that time, a new technique had come up called PCR, polymerase chain reaction. Here's your double strand of DNA. What you do, you separate the strands, you get your 2 strands, and you get look at the Watson strand up here, and you make a quick strand because DNA is complementary strands. So the Watson strand makes a Crick strand, and the Crick strand down here at the bottom makes a Watson strand. That's slang when you're talking about the two strands. You don't know which is coding or not coding. You just call one Watson and the other one the Crick. So now you've got two double helices. You melt them, you separate those strands, double them. Now you've got 4. You repeat the procedure. Now you've got 8. And you can keep doing this till you've got, you can do it 10 times, 20 times, so you can fill your whole test tube up with DNA. So he tried doing that with this ancient DNA. And lo and behold, he got some. If you look at Lane's, okay, this is an electrophoresis. What you do, you put your samples up at the top here. DNA is negatively charged because of the phosphates. You put the positive charge down here, and they all migrate at a certain speed and depending on their size. And the lanes 2 and 7 were ancient DNA, said the experimental, and sure enough, they got DNA. Well, that was exciting. It was worth publishing. They got, he was the first to get ancient DNA amplified, 750 years. Now, a couple of years later, hikers climbing this is the Finalspitze, and that's not a hike. I call that a climb. Actually, it was experienced climbers actually went up. And the Ötztal Alps on the border of Italy and Austria. So these guys are, actually it's a woman, it's a couple, a guy and a lady were hiking up, and they found in the ice, well, they were coming down and they're off the usual trail, and they got off, I don't know, just to take a hike, and they looked in the ice and they found a body in the ice, and they said, holy cow. And this was 1991. So they get down to the ground or to the civilization. They tell the authorities. So the next day, they tell the authorities come up, and some helpers, and they're recovering this body. They thought at the time the body was from a music professor who had disappeared on a hike in 1941. So that's what they thought they were working with. It turns out it wasn't. It was ancient Otzi. So they brought him down. It was kind of unfortunate. They ripped off his clothes, because that's what they do when they go to ER. People, they just cut your clothes off and take you in. So they threw all that stuff out, took him down, and they were started examining him, and they had found, he had an old axe with him, some kind of basically Neolithic axe and a bow and arrow. So they kind of realized this is not a music professor. So in any case, so they got him, they thaw him, they do an autopsy on him. Turns out poor Otzi, they call him Otzi, it's the Ötztal Mountains, so they call him Otzi from the Ötztal Mountains. Do an autopsy. He'd been shot in the back with an arrow and probably bled out pretty fast. And so I guess something went wrong up there in the mountains. He had just had a dinner of ibex, so he was comfortable, but the arrow wasn't good. And they threw out his clothes. That's probably something like Otzi looked like 5,300 years ago. Hey, another said they see his bow and arrow with him. They use his arrow, or rather his bow, as a lever to pry him out of the ice, and they broke it all up. So they weren't, archaeologists were not happy. And so they broke his bow up. Anyway, so since Brian Sykes had already gotten some DNA to amplify from the medieval cemetery, he heard about this. And so he went down there, picked up some bone fragments from Otzi after the autopsy, took it home or took it to his lab, tried PCR amplification, and sure enough, he got DNA from the Iceman. It's what they called him in the casually, he was called the Iceman. And he noticed that the sequence in the DNA matched, seemed to be pretty close to modern sequences. Our genome hadn't been sequenced, but by that time, the mitochondrial DNA, which is again much smaller and easier to work with, that had been sequenced. Now, so he starts, he says, well, that's interesting. If it matches the modern sequence, why don't I start sequencing some more people? So, and he's at a party. He has a friend of his, Mary Mosley, who's very interested in science. And he tells her all about the find of the Iceman. And he's trying to catalog mitochondrial sequences. She says, well, gee, why don't you do my mitochondria? So she donates a sample, and he types out her mitochondrial DNA, and quite surprisingly, it matches with Otzi's. Well, he publishes a note about this, and a reporter from the Sunday Times hears about it, and he inquires, and he asks, well, who is this? Who is this modern human that it matches with? And so Sykes gets back to Marie, and she says, yeah, it's okay if you publicize my name. So next thing you know, it's in the headlines. Iceman DNA matches with Lady in Dorset. And that got all over England because they're really into ancestry stuff. And it was pretty worldwide too. It got quite a media frenzy. So there's Iceman. Now, let's digress a bit. When I say sequencing mitochondria, what he's really doing, this is here's the mitochondria. These are the structural genes, the genes for proteins and some tRNAs. Right here is called the control region. It's got some hypervariable regions, as they call them, which are not as constrained evolutionarily. So mistakes can come in the hypervariable, maybe one every base every 5,000 years or so, which makes it, it's about 10 times faster than, or even higher than normal mutations creep into our genome. So it made it good for doing more shorter term dating, like on the order of 10,000s of years or 10,000 years. It's not good enough for identification. There are some really hypervariable reasons for that, but that's more forensics. Okay, so he's sequencing these control regions, and he notices that they fell into about 7 groups. And well, 95% of the Europeans he had checked, they were all Europeans at that time, they were in certain groups. So then he expands, and he does 800, he ends up with 821 sequences. Now, this is a DNA sequence you may or may not be able to see, but up at the top is the reference sequence. They call it the Cambridge reference sequence, which is the standard mitochondria sequence that they use. And below you'll see, here's a number 3 here, has a T turned to a C, and there's some other changes all the way down the line. Those are the mutation changes we're talking about. And he catalogs them all, and they're in 7 groups. And once you get different DNA sequences, you can compare them or how much alike they are. And that gives you a kind of an evolution, does give you an evolutionary relationship. So he's numbered, these are the 7, this is all of humanity here. These are the 7 sequences. I don't know what that is. Let's see if we can turn all those things off. Maybe we can. Okay, these are the 7 clones that he was using. See, if you look down here, there's a J and a T. They're very similar, so they clearly diverge from each other not too long ago, in the thousands of years, actually 10 or 12,000 for J. There's some other ones. And this, you get this evolutionary tree. X is a little higher up. And each of these goes back to one person. Just one person had the mutation and started a new line. So that's the one, those are the ones we see now. So each of these is a founder. And you can go back to here, to the out of Africa. That's about 150,000 years ago. And these people went to Europe, and these went on to Asia and on down to North America. The Native Americans are down here. So there's a whole pageant of migrations of people, a timeline, and sort of a timeline. These tree diagrams aren't perfect timelines. Sometimes I prefer star diagrams. It sometimes implies time that shouldn't be, but for all practical purposes, it's a timeline. And you can go back here, L2, this is the first in Africa. So man originated in Africa, because all these things, this whole pageant goes back to Africa, these L African types. And so presumably man originated in Africa, and that goes back to a mitochondrial Eve. There was one person that started all of this. One lady, it turns out, in this particular case, is called the mitochondrial Eve. Why do I say lady? Well, mitochondria, whereas your chromosomal DNA comes from your mother and your father, it's all mixed up. Mitochondria come are matrilineal. You get your mitochondria of your mother. Like, let's take this family down here. There are 3 people, okay, and there's a woman and 2 guys, and they all have the same mitochondria as their mother. The guy she married they don't get the mitochondria, and the mother has the mitochondria of her mother, and you can go all the way back through a matrilineal line to the first woman that had that line. That's called the mitochondrial Eve. And if you notice, on the male side, if they have children, this mother, her daughter has got the same as hers, but she marries, excuse me, oh no, this is a guy. Okay, she has a son over here. The son has got her type, but when he marries, his kids are going to be the type of the mother. I hope I explained that. It's matrilineal all the way. Kind of neat. It's very simple. It makes the genealogy stuff a lot easier when you're talking. The genes aren't all mixed up, so they're really into mitochondria for evolution. Okay, so Sykes has these 7 groups, and he gives them a name, a location, and a life. Remember I told they put letters on them, the J and T and U. Well, so he calls the U Ursula. And my previous thing had a little more information, but those slides didn't come through. Anyway, Ursula was the oldest clan. She lived about 45,000 years ago. They used stone tools. Ursula's clan members drifted all across Europe. They're now 11% of modern Europeans. This is the thing, they're all over. There's some geographical localization, but not a lot. When they first started doing this, they said, oh boy, we'll be able to localize everything. It really wasn't that simple. And we'll talk about that more. There wasn't a French DNA or a German DNA. It was really all mixed up. Maybe a Franco-German type, but that gets more complicated. And according to Sykes, this is in the book, he has a little chapter on each woman, and he talks about how they lived, what they did. Ursula, according to Sykes, lived in a cave in Greece near Delphi. She was her mother's 2nd child. The first child was taken by a leopard. Life was hard back then. You won't like it. It was hard. Lions, tigers, and hyenas ranged through southern Europe. She spent her time foraging. Okay. Now, Xena is not much is known about Xenia, but it's believed that she lived in the Caucasus 25,000 years ago. Just before the Ice Age, they spread across Europe and even reached the Americas. 7% of modern Europeans and 1%, so some of them got across the Bering and got to North America. Xenia was born in a hut made of skin and moss on the frozen tundra. I'm quoting Sykes. They had fire, but wood was scarce, so they burned bones and moss, and they were still migratory. Helena lived in the ice-capped Pyrenees between France and Spain. The climate warmed. Helena's descendants trekked from France northward to what is now England 12,000 years ago. Members of this group are now present in all European countries. It's the most successful. 41% of modern Europeans are Helena. Her father was a craftsman who made spear points to hunt bison using his new spear thrower. Helena's brother was killed hunting wild horses. They had rituals and magic to help in the next hunt. They were at 25,000 years ago, and 12 rather. Okay, now Velda was originally from Spain. Velda and her immediate descendants lived 17,000 years ago. Later, relatives moved into northern Finland and Norway. They're now 4% of modern Europeans. Velda lived in a cave in Spain. Her man was a good hunter, but he was killed by hyenas. Velda kept the fire going and was afraid of leopards. There were leopards in Europe. We don't think about that. Lions, leopard, all those things, cheetahs, all that stuff was, we think of those African. No, they were there. Her granddad was a cave painter and she died at age 48. Let's see, what happened here? Did we miss some? Okay, yeah, Velda Tara. Oh yeah, Sykes maternal, Sykes is a Tara. Sykes' maternal ancestor goes back to this group, which settled in Tuscany 17,000 years ago. Descendants ventured across Northern Europe, crossed English Channel. 19% of modern Europeans descend from Tara. Now, Tara lived at the same time as Velda, but in northern Italy. They had cave paintings and hunted deer and wild boar. They lived by the beach. Her dad made a flute for her, but died when she was 10. She made the group's first boat, which they used to go fishing. Well, and so again, Sykes' mother was a Tara, so that's what Tara did. Katrine, members of this group lived in Venice 10,000 years ago. Today, most of Katrine's clan lives in the Alps. They're now 10% of modern Europeans. Katrine, the climate was warming. Bison, reindeers, and mammoths were rare in the summer. There were bison, reindeers, and mammoths in there. Boy, but they were moving up the mountains, so they had to go further to get them. Katrina's father threw some meat to a wolf. The wolf came back for more. Then the wolf came back and had 2 cubs, which grew up with them and went out on hunts with the men. 10,000 years later, they're still our companions. Okay, now Jasmine, her people had a relatively happy life in Syria, Middle East, where they farmed wheat and raised domestic animals. Jasmine's descendants traveled throughout Europe, spreading their agricultural innovations with them. They're now 12% of modern Europeans. Jasmine lived in a village near the Euphrates in Syria. Jasmine collected acorns and pistachios. Jasmine's husband was not a good hunter. So he ground seeds, he planted some seeds and invented agriculture. Well, that's where agriculture started in the Middle East. So I wouldn't be surprised if Jasmine actually invented the agriculture.
Yeah.
I bet she did, but we'll find out. Well, we won't find out, but that's, you never know. Later, they planted chickpeas and lentils. The men brought home 2 goat kids from a hunt and raised them. And people from other villages started copying them and agriculture spread. which it did from the Middle East. And another thing, once you have, they've got dogs, then, but now when you have agriculture, that's when you need cats because you're storing food to get over the winter. So you want to keep the mice and the rats down. So that's where cats came in. You don't get cats too. You have dogs when you're hunting parties going out, that goes way back. But cats, you need to have a house to have a, okay. So anyway, so here are the 7 daughters of Eve. Here's a picture. And here's the Jasmine in the Middle East grinding something or making something. And it's kind of hard to see, but they're all doing their particular things, which and their dates. Here's another display. Here's a more geographical. Again, these are pretty well, some of them are somewhat more centered than others, but they're not mixed up. There's a little more correlation to language groups. There's a little bit of that, but they're really spread out. It wasn't, it was a surprise to see how spread out and varied things were. Now, I used to work in Clyde Hutchison's lab here in UNC, microbiology department, and we were doing a lot of cloning and sequencing. So I thought I'd prepare some of my own DNA, and I PCR amplified it and sent it to the UNC Sequencing Center. And so what you do, that's it. And before, years ago, and Sykes talks about in the old days, whoops, something fell out. No, maybe it didn't fall out. We used to do sequencing by hand. So if any of your science people, here's a sequencing gel. So you see, you put your DNA at the, anyway. Okay, put your DNA at the top, and you get the bases, and you can count up the sequence, T, 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 A, C, A, 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 A. Anyway, read the book. He talks about how to do a sequencing gel, but no one does that anymore. And because now you just send it off to the sequencing center, and you get back this sequence, and you're going to see, you see the peaks and so forth. Let's blow that up a bit. So here's a sequence. As they do it, I think T's are sort of half colorblind here, but T's are red. See what you put this at the top. What they do, they charge these things up with dyes. This is done with radiation. You have radioactive things. And you put film, after you separate, you put film on it, and the radioactive exposes the film, you develop the film. It's kind of fun, but it's a little more laborious. This, you just stick it in the machine. Each base has its own color coding, the little dye on the base. T's are red, C's are green. So if you're going to go, but here in the sequence, it's C, T, C, T, G, T, I don't know. This is, I should point like this, but you guys get the idea, I hope. Yeah, and here's, see, here are 4 G's in a row, and you can see them, G, G, G. There are 2 A's. The green's kind of faint on this one. There are 2 G, 2 A's, and then there's a G and a C. And you can read off your sequence. So that was kind of fun. So I got my sequence back, got a whole bunch of it, just reading it off. And not everybody has their own DNA sequence. Well, you can now. You can get, the docs will do it. They do it for medical reasons. So it's getting more common now. But so I did my own DNA. And I am a Jasmine. Ta-da! So that's me. That's where I fit into all this. And if you look at it, where's Jasmine's down here? She's down here. She was close to Tara here, and they go way back. And this is a star diagram or a sequence diagram. That's what I call them. Sykes calls them something else, a string diagram or something. But it doesn't give you any, doesn't make any time implications, which is why some geneticists prefer them. Here's Helena, the biggest one. And they go back to here is what they call the last common ancestor of all of people. So that's where that mitochondrial Eve. After her, Xena went off this way, some more. Here's another common ancestor. Helena went here. and they went to Jasmine, and this all goes back, as I said, to one individual, the lady. Kind of neat. And before here, you go to Homo heidelbergensis, I think is the last common ancestor between humans and Neanderthals. And that's a whole nother story. You got Homo habilis, Homo ergaster, Homo heidelbergus, and all that. And then you get to the LCA, the last common ancestor of all of humanity, the mitochondrial Eve. Anyway, they kept on going, and here's Jasmine. So here I am. Ta-da! Okay. Anything? I think there's another slide, but it's not showing. Oh, that's too bad. This must be an old. I thought this was the 31 copy, but it's not. Oh, that's too bad. Because let's see if I can remember it. Do I have it in my little booklet here? Because I was taking notes through the book as I read it. Hang on. It was the neat things that Sykes had done and his conclusions. Number one, okay, first we originated in Africa and we spread from there. At the time, you go back to the 70s, there were two theories. Maybe we all started somewhere and radiated. There's another theory called the multi-regional theory that man sort of developed all over the world. It's very egalitarian, or politically correct or something. And it turns out the multi-regionals were wrong. This is clearly, we started in Africa and spread out. And that's how it happened. Number two, at the end, this last chapter in the book is all about the philosophy or something. He said there's no genetic basis for any kind of rigid ethnic or racial classification. Mitochondrial types more or less spread evenly over Europe. Geography is more important than ethnicity. There was no, quote, French or German DNA. There's supposed to be a slide with all this stuff, but it didn't come over on this copy. Anyway, okay, number three, the Polynesian, this is one of the chapters in the book, came from the East. He was vacationing in Tahiti because once he hit the big time, got to do that. Once he got, and he said, well, why don't we check out the Tahitians? So he took samples from all his Tahitian people there at the hotel and so on, gets back. And he sequences, at that time, there was two theories. Either Phoenician Polynesians came from the East. There's a guy called Thor Heyerdahl had proposed that they actually came from the West. The South Americans made the boats and rowed over. It turns out the gene spread is definitely from the East. So Heyerdahl, in general, wasn't right. They came, but it turns out there was some South Americans in prehistory did make it West. There was some West-East, but not contributing much to the gene pool. There was the genetic flow was West-East. So Sykes was right. Heyerdahl could have maybe a little bit, maybe a few pontoon boats over. Okay, what's the next thing he did? Okay, we studied Iceland. Iceland, now this is really interesting, I think. If you look at so we do the mitochondria, you can, after all this, then they started doing it for the Y chromosome, which is paternally inherited, because clearly all males have the Y chromosome. There's an X and there's a Y. So Y is all the men in here have their father's Y, all those fathers have their father's Y. Has nothing to do with the women. Okay. So they start sequencing the Ys, and he finds out that the Ys came from Scandinavia. Okay, well, that was nice, about 75% of them. But to their, not surprise, as the stories were right, most of your mitochondria, the female came from, they call them Scottish or British Irish. There's a British Irish group. And why was that? about 66%, because basically the Vikings would sail over, get to northern Scotland. This is in a Nature article, called them coastal raiding parties. I don't know if they're partying on, but they would live, I guess they would kidnap women and take them to Iceland. So to live in Iceland. So very interesting genetic story of the development of Iceland. That again, the men have Scandinavian Y and the women have, they call them Irish British mitochondria. Okay, Anna Anderson, another thing, he did the Romanov, they found the Romanov bodies, but even before that, he had been interested in it. And turns out Anna Anderson, they had, who claimed to be Anastasia all those years, and they, Ingrid Bergman got an Academy Award for playing Anastasia. So that was a great story, but it turned out in the 70s, she'd gone to the she died in 84. And in the late 70s, she'd gone to the hospital and had a biopsy done for some reason. And then we're up to about 1994. And it turns out the biopsy is still there. This is probably before HIPAA, because you can't do that now, but back then. So they let him look at the biopsy. Turns out, no, I'm sorry to disappoint you, Anna Anderson was not. Now, some of her defenders say, oh no, the biopsy sample got confused, but turns out that DNA matches to a Polish woman, possibly a courtesan of Nicholas II, and that's why she kind of knew all what goings on, which is going on in the royal house. So she got to impersonate Anastasia. Interesting. Okay. And another thing, he sampled. This is for the fun. He's in the Bigfoot and Yeti. So he starts sampling. They're in museums and private collections. They say, well, he sampled. They're mostly bears. Bigfoot and Yeti, mostly some wolves, some human, one ungulate, but no primate. Some human, but that's the only primate. There weren't any other weird primates or something. So Bigfoot's looks like they were all just known other animal species. And oh yeah, so anyway, I guess that's about it. How is we for time? Should we ramble on or 2:24? And remember, Jurassic Park was 1993. So once the Iceman thing came out, that was really big. That was everybody. The phone is ringing off the hook. People wanted to clone Otzi or to clone themselves. Or it was crazy. Anyway, so yeah, think of anything. Let's see, I might know something. I don't know. Hey, what's Andres? Yes.
How long?
Oh, yeah. Not yet.
Is there extra mic here for people to ask questions?
We're going to pass the yeah,
That's the actual. This is for the audience. This is. Stand up to her.
Oh.
Okay, our society is an all volunteer organization and we receive no money from any other organization. So we survive due to your commitment and your generosity. So please share. And what we'll do with the questions, if everyone will raise your hand and Chris will take the mic around to you and ask only one question at a time. And Mike can come and answer.
Mike, put the mic on your shirt.
Yeah.
When you had your mitochondrial sequence taken, how long was it? How many pages and how many base pairs?
That one run is probably about, you get about 400 bases of good sequence. You can look at this. It goes through 20. This is 410. I didn't have the second page. They start getting, once you hit about 500, you get a lot of N's because the electric eye can't read very well. They get all spread out. So I'd say 400, you get 400 on a run. Then you get, it goes out to about 800, but it's too many N's. N meaning nucleotide, meaning they don't know what it is. Because X is something else usually.
So the next is not a question, but so that you all know that you pronounce the umlaut in Etsy. It's not Otzi, it's Etsy. 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 It's Etsy umlaut. It's German.
Yeah. It's more like Ötzi? No.
Etsy.
Etsy. Okay, sorry. Meine Aussprache ist nicht
Ja.
so gut.
Mike, are all these daughters, they're the samples of the DNA that were taken? There's actual remains that were found or not?
No remains of any daughters have been found. What they do is they sequence the people here now. They're doing more and more archaeological samples, and they typically fit in with what they find. But the actual mitochondrial Eve is not found. She's hypothetical.
Okay. Oh, so they just go by the changes that they see over.
If you're making a family
For the whole
tree,
population.
it would go all the way up to this mitochondrial Eve. We don't know.
Okay.
There is one. We know there should be one, but we really don't know who she is. It's 10,000, 20, 40,000 years ago. There's
Okay.
no trace that we know of now.
It-
And they didn't know either. And there were other women with them who may have had other types that didn't make it. There's another thing that one of the first things he did was the Syrian hamster story. He had read in the papers that all the Syrian hamsters came from some hamster 100 years ago or something, all the hamsters. So he checked them out, and sure enough, they were all related. And if you did the genealogy and the timeline stuff, they all went back to one Syrian hamster. And that's been found for lab mice. You go back far enough, you get to one person, and all the brothers and sisters they had didn't, unrelated people didn't make it. You can go back far enough to one founder. And with the mitochondria, you call them the mitochondrial Eve. You can get the same with the Y. They call them the Adam Project. That came after the Eve Project. They call them Y chromosomes. You look for the Y Adam, and they have a number of different groups. It's quite similar, except Y is not as easy to work with. It rearranges a bit more and stuff like that. But yeah, there is a Y project.
I just have a quick question. Because I've done one of those like home ancestry kits.
Yeah.
When you have that diagram of all the different Eves, is that also what they consider like a haplogroup? Is that the same thing? Because it'll say like LE and then it'll have a whole bunch of numbers.
Well, that's not your chromosome. That's your haploid chromosome. That's
Okay.
the 3 billion bases. Typically, you have 2 chromosomes. And they're supposed to be the same, more or less. They're pretty much the same. So you sequence one or the other. But the haploid number is 23. It's kind of weird. It's not 4. It's the haploid is just chromosome 1. Diploid is 1 and 1. And it's you have 2 of every chromosome. Diploids 46, haploids 23.
I'll hold for it. Okay.
Mike, presuming there is one unique ancestral Eve, you're also talking here about 7 different faces of Eve and maybe, others that, that add up to 100%. How do you account for that? Mutations, or why is there not just one unique mitochondria?
They all go, let's see, the European ones are like 10 to 40,000 years ago. And there was, it goes back to one, that's called the most recent common ancestor right here. All of the other people who lived there did not leave any living descendants. It's a hard world with leopards, tigers, hyenas, but all of the people, none of the people here, or none of the females left any living descendants except for this one lady. If you go far back enough, you can get the Eve. Yes,
Okay.
it's astounding. It's weird if you think about it. But all of the, now there weren't a lot of people. There was called the bottleneck around this. There was a bottleneck in Africa 200,000-300,000 years ago. There might have been 1,000 people, something as few as 100 or 200 mating pairs. And that was why we're all the same. If it was a big population, there'd be a lot of variety, but it was a very small, very homogeneous population at that time, and almost went extinct. A lot of others, Homo habilis is gone, Ergaster is gone, Heidelbergensis is gone, habilis is gone. But we're still around. We had a tough time a couple 100,000 years ago.
One last thing then, is somebody trying to do the study on, they've got some Neanderthal DNA.
Yeah.
Have they been able to try to do this using what they've got of Neanderthal DNA?
I don't know a lot, but they've got a Neanderthal ancestor, is probably this Heidelbergensis, maybe 400,000, 500,000 years ago. He's been sequenced mostly. Techniques have improved. They found a toe bone or something, and they got some good DNA, enough out to get most of the sequence, 90, I'm sure in the 90s. When they say sequence, sometimes not 100%, but it's pretty high. And he's been sequenced. Several of them have been sequenced. They've got, I'm not up on the whole my Neanderthal story, but yes. Pardon? Say it again, Bob. Say what?
You don't know if Neanderthal will be one of those 7.
No, the Neanderthal was way back here. See this first, it's a totally different branch. You see this, all the 7 sisters came from this common here. You go way back to over here is the common ancestor to Neanderthals and people. Neanderthals went this way, and there are other, they've got a number of different Neanderthals sequenced, and they're putting all that stuff together. Let's go, come on. But that's way back, and they went this way, and we went that way. I'm putting the most recent common ancestor of humans and Neanderthals is Heidelbergensis. That's maybe 400 or 500,000 years ago. They went this way, we went this way, they went that way, okay. And then we get to this point, all humans go back to this person. Now this person, I said, there were other people living in the group. There were others, but none of their lines survived. Well, I know exactly.
But let's-
This is
let's-
95% of your.
We have another
There
person who
are
wants
some
to
minor.
ask a question, so let's continue to ask questions from people who have not had a chance yet.
Where do the Eurasians of Central Asia and Parmids, what is their ancestry? Because ultimately they moved east and west and do the
Say-
dominant.
Start over again, please.
The Eurasians
Yeah,
in the Central Asian plateaus, who ultimately went east and west and became the dominant species. What is their ancestry? How do they trace back their ancestry?
well, the Asians, let's see, there was this genealogy here. This is out of Africa. So they started in Africa, and basically Europe is the group. The Europeans are here. There are other European, there's some minor groups. When he says it's 7 daughters, there are other ones and they may find more, but this is 95%. The ones with the red are the 7 daughters. 95% of Europeans came. This is sort of oversimplifies things. 95% of Europeans came from here. There's others. If you go back to the most recent common ancestor of out of Africa, okay, and this went here and this line went here. And this is M, I think, is one that seemed to go more toward Asia and cross the Bering. I'm not a population geneticist, something like that.
I just wanted to share an anecdote, and I'll try to be quick, but it's sort of like every advance in technology has interesting ramifications. So, you know, everybody has been doing 23andMe for a while or the other one, and a bunch of my family has done it. And this summer, or one recent trip up to the Catskills, my cousins were talking about this mystery cousin that we had, because several of them who did, it wasn't 23andMe, it was Ancestry.com, which of course the Mormons are big into that. But they said, we have this mystery cousin named Gary Stiles. I had a name for him. He was popping up on all, we had a mystery cousin who was popping up on my cousin's profiles, and they were joking about it. And I happened to mention it to my brother, who has also done Ancestry.com, and he's coming up on my brother's profile as a brother. So I don't really care about this. I haven't done it. I haven't done my DNA, and I don't intend to do my DNA, but it makes me really angry that there's this big corporation that is looking, you know, is able to put all this together so that a bunch of my cousins think we have a mystery cousin. My brother is horrified that we have a potential mystery brother, and he's sworn me to secrecy, don't talk about it, and I don't intend to. But, you know, the idea is my dad's probably the culprit here as opposed to my mother. But, you know, the ethics of all of this is very interesting. And again, to me, I don't care. I just don't care. But my brother's really upset about this. And what right does Ancestry.com have to potentially impact people with stories like this?
I don't fully understand upset about.
My brother is upset that he has a mystery brother and that.
Oh, yeah, look, you start doing these things, people find false paternity.
Oh, yeah.
All kinds of crazy stuff comes out. Oh, you bet.
Yeah.
Yeah.
I mean, to
Yes.
me, you can't put the genie back in the bottle. So it is what it is.
It is what it is.
But it does bother me that there's this for-profit thing
We
behind
live
it
in a
all.
capitalistic world, and it happens, and it's great that it happens this way. And mistakes can happen, and there is an error rate. You always double check everything. It's great that if you're in China, if you're a Uyghur, or you're a minority, they're going to take your DNA, but they'll tie you down and take your DNA. They're now doing all children now in China, and there's no privacy. It is specifically, they call it the Golden Ancestor Project or something, but it's specifically to be used against people to make sure it keeps everybody in order, okay? If you want to go to China, they're going to take your DNA, and there's nothing you can do about it. And they'll do what they want to, the government, okay? There's some politics there.
Every lie and deceit will eventually be found out. You want to say that again, sir?
I think everybody heard that.
Well, the people on Zoom can't hear you. When
Oh, Zoom, okay. No, every lie and deceit will eventually be found out.
Say again, every line?
Every,
I'm going back for a second. Every line.
And deceit.
Oh, lie and every lie and deceit. Is that what you're saying?
Every life.
Lie, lie, L-I-E,
L-I-E. Every
Lie.
lie.
and deceit.
Well, I don't understand.
Okay. Any other questions? All right. Thank you. All right.
Anybody else? Okay. That's the community story.
Excellent, very interesting.
Wow, I found it interesting.
Yes, we did. All righty, thank you, Michael, for a very wonderful talk, and that sounds like a book I need to put on my list. And let's see, now we have time for announcements. Does anyone have any announcements? I'd like to thank, oh, yes, okay, Dorothy. Oh.
And nobody had put it on hold. I've got it out of the library and they auto renewed it. So nobody's put it on hold. So I will return it, but I just wanted to encourage people to read the book.
What's the book this time?
Well,
Oh,
that's a good question.
is that,
But it was,
Greta, it's Greta's pick. So Greta, Celestial Bodies. It's a fictional book about a family in Oman. And so it's a really nice introduction for me anyway into that part of the world.
and I'd like to thank, who else? Oh,
I don't have a question, but what's the query? Is next Sunday's meeting in here or only Zoom?
I think it's
Hybrid, that is.
okay. And unfortunately, we don't have Solomon to give open and closing words. I should have, as I said, I should have reminded him. Though I don't know that he ever agreed. I sent him the schedule. But anyway, I want to thank Amy for being our greeter, and next week we'll have a different MC, but and Fred and Maura are off on a cruise, so we'll hear about that when they get back. But otherwise, our meeting is over, but our fellowship continues. And we don't have refreshments, but I hope they will return soon. But give a warm greeting to the person sitting next to you, and we'll see you next week.